My name is Rashmi Halker Singh, and I'm the online and social media editor of the Headache Journal. And I'm joined today by Dr. Amynah Pradhan, who is one of the authors of this year's Wolff Award manuscript entitled Migraine and Peripheral Pain Models Show Differential Alterations in Neuronal Complexity. Welcome, Dr. Pradhan.
Hi, nice to be here.
Well, first of all, congratulations on behalf of myself and the editorial board at Headache. We're so proud of you and your colleagues for winning this award. I had a chance to read the manuscript, and it's really amazing work what you all have done. So congratulations on that.
Oh, thank you. We're so excited. Yeah.
I think the study's really elegant. I think, it comes up a lot in clinical practice. People want to know about what is migraine and what's going on in the brain when people have a migraine attack, and how is that different from other pain conditions. And you have done such an elegant job really diving deep into looking at that question. First of all, can you explain to our viewers really, what was your study about? What did you all do?
Yeah, so this was a study where we really wanted to try to understand how neurons in different brain regions change in different animal models of migraine, and then comparing and contrasting it with another type of functional pain condition called complex regional pain syndrome. And so it's a follow-up to work that we had previously published, where we'd shown that in a couple of different models of chronic migraine, that there was decreased neuronal complexity in brain regions that regulate head pain. So things like the trigeminal nucleus caudalis and the periaqueductal gray. And so this study sort of followed up on that and looked at different brain regions that are involved with pain processing. So things like the thalamus, as well as regions that are not so directly involved with head pain processing. So things like the amygdala and the striatum. And so what we found was that in general, in one model that we use, the nitroglycerin model, we use this known human migraine trigger, nitroglycerin, that causes a type of head pain in mice. And what we found was that in this model, that there was increased neuronal complexity within the thalamus, and no change in regions that are not as involved with headaches, such as the amygdala or the striatum. And so that was kind of an interesting finding, because it suggests that in this like brain region that's sort of a relay for pain, that that the neurons have these increased neuronal branches and are more able to make connections because they're just a little bit bigger and more spread out. What we also found was that in a model of migraine aura, that there's actually a decrease in neuronal complexity in a region called the periaqueductal gray. And that's sort of interesting because, when you think of migraine aura and we think about this particular model, which involves activation of the cortex, you sort of think, oh, well, aura is not necessarily, associated with pain. You're stimulating a part of the brain that's the cortex, which is pretty far away from the periaqueductal gray. And so it actually was kind of interesting to find that we see these changes in this sort of midbrain region that's involved with pain processing, because it does sort of strengthen this idea that maybe these changes in cortical activity that are thought to precede migraine pain may actually then be feeding into these pain networks that could then, maybe promote this pro-migraine, pro-headache type state from the aura state. And then the last little piece of, work that we did was looking in a model of complex regional pain syndrome, which is a peripheral pain model where there's a very intense type of pain that develops often after sort of casting. And this model also, we examined a time point that's when a chronic pain state has been induced in this model. So we could really compare it to our chronic migraine model. And what was interesting is that we found that there was a decrease in neuronal complexity in a place called the hippocampus, which is, we sort of confirmed findings from another study. And this region is thought to be involved with sort of the brain fog that's associated with this chronic pain state. And the fact that there's decreased neuronal complexity there, I think is really kind of interesting. And it was nice for us to confirm that finding. But then interestingly, when we looked in this periaqueductal gray region, this region involved with pain processing, we actually saw an increase in neuronal complexity, which is in contrast to the migraine models that we looked at, where we found a decrease in neuronal complexity in that same region. And so what's overall kind of interesting about that is it suggests that in these different chronic pain states, that changes in neuronal complexity seem to be a hallmark feature of that sort of chronification of pain. but that depending on the type of pain you're looking at, there are different types of changes that occur, and that those might kind of give you a hint of which one of these brain regions might be important for the chronification of that pain and how those circuits might be altered in those states.
So I think that's really fascinating work and really validates the fact that migraine is a true neuronal brain disease and distinct from other pain problems as well. And also highlights that this is a really intricate and complex problem. It's not simply just a headache, and it's also very different from other pain conditions too. So as I found that really interesting, as well as a level of detail that you were able to ascertain, including, like you mentioned, the thalamus, which we often think of as a relay center, but you're able to demonstrate the increase in neuronal connections there, as well as the involvement of the periaqueductal gray, which we speak about a lot when it comes to migraine. And I thought it's really interesting about how your study compared that to the other pain conditions that you mentioned too. Were you surprised by any of these findings that you came across?
Oh yeah, we were super surprised. And I think, what I think the thing is that when we think about neurons in the brain, we often have this image of that's very two-dimensional, because when we look at neurons in the brain, we often tend to just see them as from the slice of the brain. We see them fixed. We don't actually see them moving and changing, right? But in fact, neurons and all cells in the brain are constantly moving and changing, right? They're responding to their environment. They're putting out projections, they're making synapses, they're retracting synapses. It's happening all of the time. And these neurons are always in this dynamic state. And so I think it's really fascinating that in a migraine state, we've previously shown that there's this decreased neuronal complexity in these head pain processing regions. And now that perhaps there's an increased neuronal complexity in the thalamus. And so it sort of suggests that, maybe these changes in neuronal complexity are sort of helping to facilitate chronification, because now all of a sudden in certain brain regions, you've got neurons that might be kind of stuck in one state or not as able to respond to the stimuli around it. And so therefore you can see how that might result in maladaptive responses, because maybe there are synapses that are formed that are just constantly firing that should have been taken down a long time ago. Maybe there are other synapses or connections that should have been formed that would have been preventive, but now can't form because the cell is kind of stuck in this shape and can't change as much as it used to. And then maybe in places like the thalamus, where there's increased neuronal complexity, it's that, oh, well, now all of a sudden the thalamus is making these connections with things that maybe it's cells that it shouldn't be making connections with, making synapses that are overstimulating other cells, which then might help to facilitate that pain chronification. So for us, it was just really cool to think that, oh, wow, these cells are changing. They're changing all of the time. And that, maybe their inability to change or their ability to change too much then can just cause these types of chronic states.
As you've just brought up, with any good research, findings always bring up new questions that need to be answered. What's next here? Where do you see the next step for this project?
Yeah, so one thing that we found that was interesting was that if you can affect those neuronal dynamics, either directly by affecting the cytoskeleton or indirectly through anti-migraine treatments, for example, that you can restore that neuronal complexity. And so I thought that was really interesting, because it suggests that there's this relationship between altered neuronal dynamics, and then if you can fix that or restore it, that has this sort of anti-migraine effect. So you sort of normalize those cells back to how they should be. And we've shown that using CGRP antagonists. And I think what I would really like to do is look within our migraine models whether other types of anti-migraine medications can cause the same thing, or is it something that's just unique to CGRP receptor antagonism? And then also see in a CRPS model whether things that are known to be effective in that model do the same thing, where all of a sudden these alterations that we've seen in the PAG or in the hippocampus are then reversed if we use known pain therapies, because that would sort of validate it. It would sort of tell us that, yep, it looks like that those changes in neuronal complexity are sort of a signature of that type of chronification. And if we can block that or restore it, we then, it sort of is a feature of, okay, yeah, looks like the brain is getting better and therefore no longer in this chronic pain state. But I also think that it's a really cool opportunity to kind of dig into novel mechanisms, because, if you can then directly restore those changes in neuronal complexity, either directly targeting like cytoskeletal elements or things that have to do with tubulin dynamics, then you've got a whole new class of medications for the treatment of chronic migraine and for chronic pain, because now you're looking at something that's very different from the sort of standard migraine type therapies that we currently use.
Well, it sounds so exciting, and I think there's so much on the horizon yet to come. So congratulations again on this award.
Oh, thank you. Thank you very much. I really enjoyed this talk.